Okay guys, so today we are going to finish meiosis. We already went through meiosis number 1 last time, which we started with interphase. We did the G1, the S phase, the G2. We went through meiosis number 1, and now we're going to not enter interphase again. We're going to go straight to meiosis number 2. So we are going to start from starting at telophase number 1, we're going to go right into prophase number 2. Okay, so big thing with prophase number 2, guys, we have a spindle apparatus forms. Nothing too crazy here other than the normal prophase activities. All right. In metaphase number 2, I'm going to draw this out for you. So if you remember metaphase number 1, we had something that looked like this. So this is, I'm just going to call it meta 1. We had our tetrads lined up in the middle. And pretty much our chromosomes were on top of one another. And then we had our spindle fibers connecting to our tetrads. And then the tetrads got pulled apart during anaphase number 1. Now metaphase number 2 actually looks a lot like regular metaphase, because we're going to have our individual chromosomes lined up in the middle. So now we have our X's there, we have our X there, and obviously, you know, during prophase number 2, we can assume that, yeah, they're going to get pulled apart. Okay? So there you go. Don't forget here, guys, when they get pulled apart, too, don't forget that these chromosomes are not going to be identical because of crossing over. So when it says they are no longer genetically identical, why? That's because of crossing over. All right? So let me go back a little bit. All right, if you remember, when we have a diploid cell, diploid means we have 2 copies, okay? At the end of meiosis 1, when the cells, you know, the chromosomes look like this, or we could say that if we have a cell here, okay, we're going to have this type of chromosome in one, and this type of chromosome in the other. I'll use blue, why not? Okay, there we go. All right. Because of crossing over, guys, this green one on this side, which I'm making slightly blue now, is not going to be the same as this one on the right side, okay? So let me draw a line down the middle. This one over here is not the same as number 2, even though they're both green. Why? Because crossing over happened and some genes got exchanged between them, so they are not identical chromosomes. And the same thing with the blue over here. Let me throw some green in there, or I'll even throw a red. Let's just to be able to distinguish. Draw a line down the middle. Number 1 is not the same as number 2. So at the end of meiosis 1, we actually say it's a haploid cell, which is very odd because we actually do have, you know, a copy still. The chromosomes are still copied, but the problem is they're not identical chromosomes anymore. So we can't call them diploid. We have to call them haploid. All right. Which, if you think about it, at the end of mitosis, you know, we still have our diploid cell. But this is just a weird terminology that I wanted to make sure I went over with you guys, because it is weird to think about. And the only reason it's considered haploid is because these two, number 1 and number 2 on both of these chromosomes, they are no longer identical because of that genetic variation that crossing over makes. Okay. So moving on to anaphase number 2, we are going to pull all of those chromosomes apart. Just make a few different ones here. So we had them like this. And then we are going to, so that was our metaphase. I'm going to pull these guys apart. Okay. And the kinetochores get reeled in closer to our centrioles. All right, so here is our anaphase 2. Now, we're later going to, in telophase, split this in half and it's going to form 2 cells. All right, but don't forget, guys, there's 2 cells at the end of meiosis 1. Since there's 2 cells at the end of meiosis 1, yeah, this is happening in one of the cells, but I'm not showing you that in reality, there's another cell somewhere doing this exact same thing, okay? So this will split in half, we get 2 cells here. This one will split in half, we get 2 cells here. So this would be our third, this would be our fourth. So in reality, we are getting 4 cells out of this. I'm just not showing you the other cell on what's going on with it. I'm only working with 1 cell here just to save some space, all right? So that is anaphase number 2. Our chromatids, our sister chromatids, have separated. And don't forget, guys, the sister chromatids are no longer identical because of crossing over. And then lastly, we are going to have our new cells form from telophase 1. Everything's going to be split up in cytokinesis, and we are going to reform that nuclei. So now we're going to have 4 cells altogether, okay? So we're going to have 4 cells at the end of meiosis 2. At the end of meiosis 1, we only had 2, but then both of those cells later divided again, okay? So those cells are still going to be haploid just like they were at the end of meiosis number 1, and they're all genetically diverse. None of them are going to be the same, all right? Okay. I think we're good here. We went over all that. Here is a comparison between mitosis and meiosis, all right? So if you look at, metaphase and then metaphase 1 of meiosis, which, think of how I worded that, guys. If I'm just talking about metaphase and I'm not putting a number after it, I'm automatically referring to mitosis, all right? If I put a number after it, 1 or 2, then I am automatically referring to meiosis. But look at the difference in metaphase. We have on our metaphase plate in the middle here, for mitosis, everything is lined up, okay? Our sister chromatids are lined up. Over in metaphase 1, we have the tetrads lined up in the middle. Now, they would overlap a little more than it's showing there, but if we overlap perfectly, you won't see all of them, all right? And then at the end, we get 2 cells in mitosis that are both diploid, and in meiosis, at the very end of meiosis 2, we get 4 cells that are that are haploid, okay? So there's your comparison between the 2. There's a little zoomed in on it. We're not going to worry about that. And then here's just a summary on everything we went over on what the differences are. You guys can look at that on your own time. Okay, the one thing I wanted to mention, guys, is whenever our chromosomes are held together during metaphase, there is this protein called cohesin, and sounds like cohesion, sounds like things sticking together. So there's a protein called cohesin that sticks these chromatids together, all right? What we're going to do is bring in another enzyme, or another protein, we can say. Specifically, it is an enzyme, and it is called separase. So the cohesin is going to be broken apart by separase, so that way our chromatids can separate, all right? So that's the relationship between cohesin, which holds it together, and then when separase comes in, it breaks that cohesin, so the chromatids can move. All right, we're good here. Okay, so last section. This one's a short one, guys. So we can have mutations, and not all mutations are bad. A lot of people think like, oh my goodness, you have a mutation, you're going to get cancer. No, that's not always the case. So we have alleles, and we'll talk about alleles later on. But what an allele is just like an alternate form of a gene. So we can have different genes. Let's think of even like your earlobes. You can have a gene for dangling earlobes, or you can have a gene where your earlobes are connected, minor dangling. But some people's earlobes are connected and they don't have a little dangle. Okay, those are just two different alleles, two different forms of a gene. When we have a mutation, we can actually make new alleles. All right, so we can have, maybe I'm just making this up, but we can have dangling earlobes, we can have connected earlobes. Maybe a mutation occurs in one of those alleles where it completely changes it, and now we have a spike at the bottom of our earlobe instead. Okay, so just a mutation. Some of them are good, some of them are not good, but most of the time, guys, mutations, they don't really play a huge role. But every now and again, a mutation can change and it doesn't get caught, and then it changes whatever it's going to change, and that's how evolution occurs. So 3 different things we're going to go over, and this is going to try to contribute to genetic variation. There's independent assortment, there is crossing over, which we already talked about, and then random fertilization. Okay? So I'm going to draw a picture of independent assortment here. And this is going to be with meiosis only. Okay? So this is in meiosis. All right. So let me draw my cell here. So this is going to be between metaphase. We'll do metaphase and anaphase. All right. So during metaphase, we could do metaphase number 1 if we want to be real specific here. Okay, let me draw my chromosomes lined up. They're going to be in tetrads, obviously. Okay. So what's going to happen here, guys, is they're going to assort independently of one another. So all this says is, let me change my color here. For independent assortment, okay, we could have, I'm going to draw it as 2 new cells here, guys. It'll make more sense this way. I forgot to draw my number 1 there. There we go. We could have this one here come to this side, and the other one come to that side. Okay. What's going to happen here is these traits are going to assort into the different cells independently of one another. Reds are going to go to one, greens are going to go to the other. Now, it doesn't matter which ones we put in where, as long as we have one in each. That's independent assortment. Okay. So that's pretty much all you guys need to do or need to know for independent assortment. Okay. For this one, guys, we are going to have a lot of different ways all of our chromosomes can assort from one another. So if we have, now we have 23 different pairs, okay? So we have a lot of different combinations of things. If we lined all 23 pair up, and I'm not going to make all the colors here, but I don't have enough color to do all of them. Okay. And I'm going to add, we'll make this one a little bit black on this side. Make that one. Okay, I'm just kind of showing crossing over with different things. We could have this one go to this side, this one go to this side. On this one, maybe we have this one go to this side, and this one goes to that side, okay? And on this one, we could have this one go to that side, this one go to that side, okay? It doesn't matter which way they go, but with 23 different chromosomes, we actually can have, if you do 22, because we have two different alleles, or two different chromosomes. If we do it to the 23rd power, because we have 23 chromosomes, we are going to have a lot of possibilities there, okay? So that 23 is just our number of chromosomes that the individual has. So for us, if we have, 23, you would just do 2 to the 23rd, and that's how many combinations you could have. If an organism has only 16 chromosomes, you would do 2 to the 16 power, and then that would determine how many different combinations you can have in the gametes. In other words, making those sex cells, making the sperm, making the egg. All right. So independent assortment, it's nice, guys, because that also influences genetic variation because we have different ones going to each side, right? And no two of your sperm cells or egg cells are going to have the same combination. So you guys can see here, we have a bunch of different combinations that they can be right there. And this is only using like 2 chromosomes. Okay, second one, anytime you see recombinant, that is simply just different than the parents. That's what we mean by recombinants or recombinant chromosomes. So crossing over is going to give us DNA that is different than our parents. And we already went over crossing over, but here is another picture of it. You can see that there's the chiasmata there. We are going to have that area, and then we are going to have a split between those two areas. So we're going to have crossing over in that areas. Hopefully the same genes get transferred. Okay, we want to make sure we have the same genes on each one. In other words, we want to make sure that on one we have maybe the gene for, I'll use eye color because I think I used that in my previous example. The gene for eye color is on the one, gene for eye color is on the other. We want to make sure those areas cross over and that we don't have like 2 alleles for gene color on the one and none on the other, right? That would be bad. And then also, we want to have random fertilization, all right? In other words, we don't want to like pick out the sperm and the egg that are used. It should be random, all right? So given that we have like over 8 million possibilities, okay, there's a lot of variation that could occur there. And we saw that through independent assortment. All right, so last part, guys, just goes over evolution. Natural selection results in the accumulation of genetic variations favored by the environment. So based on mutations that happen, guys, now they might be mutations, they might just be genetic variations. Some organisms are going to be better adapted to that environment than others. Those organisms that are better adapted will probably survive and thrive, and the ones that are not, well, they probably won't reproduce, okay? All right. And I think that's it for the chapter, guys. Yep. Okay, so that's it. Keep working on your chapter 10 assignment, guys, and I will talk to you later. Have a good day.